Hi, welcome to my presentation. My name is Greg Taylor from Lab 16, and I'll be presenting my findings on halophilic archaea and the possibilities for extraterrestrial life, specifically on Jupiter's moon, Europa. Alright, to begin, some background knowledge on archaea would be beneficial. We'll start with the origins of the domain. Archaea are single-celled prokaryotes and are both extremely old forms of life and relatively very young too. So their fossil record dates back to 3.5 billion years ago, but they're only introduced as their own domain 44 years ago. So in 1977, microbiologist and biophysicist Carl Woese noted enough distinct characteristics that differ from the domains eukarya and bacteria to make archaea its own domain. So one of the main different characteristics is the habitable environment for archaea. Archaea can live in environments that other organisms would have no shot at living in. And this quality makes archaea referred to as extremophils. Some of these extreme environments include hydrothermal vents, terrestrial hot springs, high pressures like up to 10 kilometers deep in oceans, anaerobic environments, acidic environments, highly saline environments, and even toxic sludge, which is what this photo on the left is. So pretty extreme on all counts in all environments that both eukarya and bacteria species could never survive in. The specific environment we are interested in today is the highly saline environment. Extremophils that can survive and thrive in these highly saline conditions are called halophils. So just to lay it out, halo is Greek for salt and phile or philic for loving, so salt loving. On top of being able to live in high saline environments, halophilic archaea can survive desiccating conditions, starvation, and radiation. So they have these mechanisms or practices to help them survive, and all are really quite impressive. So to survive nutrient-poor environments, these single-celled prokaryotes alter their surface area to volume ratio to maximize surface area, which leads to maximizing nutrient uptake. They also have the ability to rebuild whole chromosomes from fragments, which allow them to survive radiation. And their ability to live in high saline environments depends greatly on their metabolic pathway known as the methyl aspartate pathway, and this limits osmosis and allows the halophiles to retain their water. The halophil examined in this experiment is extraterrestrial. It was isolated from Jupiter's moon Europa, which is pictured on the left here, and Europa's environment is very extreme and most likely only a place an extremophil could live. The moon is a complete ocean of salty water with a surface of 15 to 24 kilometers thick ice surrounding it. Being so far from the sun, the surface temperatures are very low and there's very little light. And of that light that reaches Europa, most of it is reflected by the icy surface. So we can assume that because of this low light, there's going to be very little nutrients. The halophile was isolated from the subsurface ocean 1 kilometer below the ice crust of Europa's Cilix crater. The water sample the isolate was contained in had high concentrations of sodium and magnesium salts. And just really briefly, the importance of investigating archaeans. Understanding the environment where life can occur, including environment off of Earth, is beneficial to our understanding of extraterrestrial life in general. So understanding how extreme environments are tolerated can lead to manipulating how organisms other than extremophils can survive there. And finally, investigating how life can be found on Europa can lead to the discovery of more life outside Earth. So the purpose of this lab is to compare and contrast an extraterrestrial isolate from a halophil found on Earth. This way we can determine if the isolate is halophilic. And we'll do this by examining the morphological, biochemical, and physiological characteristics of both an organism from Europa as well as an archaea from Earth. And that one is Halobacterium salinarum. So by the end of this presentation, hopefully we'll be able to see the similarities of the two species and also consider the possibilities of terrestrial archaea living extraterrestrially. So now I'll just go over the methods that were used for each part of the comparison between the two archaeans. For the morphological characteristics, photographs of both specimens were taken and qualitative observations for each were recorded and compared. These observations included colony size, pigment, density, consistency, and more. For the physiological characteristics, we were interested in the amount of growth the specimen showed while we manipulated the environment they would grow in. So 20 microliters of liquid culture was inoculated into 5 milliliters of growth medium in each case. So to test the effect of sodium chloride concentration on growth, the absorbance at 600 nanometers was measured after 2 weeks of growing in varying concentrations of 0 to 6 moles per liter, but temperature and pH remained constant during that. To test the effect of temperature on growth, the absorbance at 600 nanometers was measured after 2 weeks of growing for varying temperatures of 5 to 60 degrees Celsius, but sodium chloride concentration and pH were constant. And to test the effect of pH on growth, the absorbance was measured for the culture growing at pH ranging from 3 to 9, but all other variables kept constant. For examining the biochemical characteristics of each Archaea species, the presence of enzymes and acids were detected. So acids can be produced as an end product of carbohydrate metabolism, and we tested whether the two species produced these acids by growing the culture in media containing carbohydrates and using phenol red, which is a color-changing indicator to show the presence of these acids. So a similar approach was taken to determining the presence of enzymes. The cultures were grown in media containing the molecules the enzymes would break down, and color-changing indicators were used to detect the presence of those enzymes. And finally, the sensitivity that both Halobacterium salinarum and the Europa isolate had to different antibiotics were determined. So this was done by growing the cultures in growth medium containing these antibiotics and recording the effect on growth. These results were recorded as sensitive, intermediate, or resistant. All of these observations and results were recorded for the two archaeans and compared with each other. So in the morphology section, the idea is to begin characterizing the halophils and comparing their physical qualities. So the Halobacterium salinarum is pictured on the left, and the isolate from Europa is pictured on the right. And I've just outlined some trends regarding similarities and differences between the two. So form is the overall shape of the halophil, and we can see from these pictures that they are both circular. Another similarity is both their margins are entire. Alright? And the margin is how the edge of the colony appear when viewed from above. The fact that the colony completely reaches the edge of the circle makes these halophil colonies entire. There are some distinct differences with these two colonies though. From the pictures, we see that the colors are different and therefore have different pigments. The Halobacterium colony being a light pink color and our Europa colony being a light brown color. As well, these two colonies have different densities, and this is determined by how much we can see the background through the colony. So the Halobacterium salinarum is completely opaque, and we can see a little bit of the black background coming through the edges of the Europa isolate colony, so it is semi-translucent. Okay, on to the biochemical test results. The first test the colonies underwent was the presence of acid as a product of carbohydrate metabolism. So the colonies were grown in 14 different media, each with a different sugar, and the results of the presence of acid production between Halobacterium salinarum and the Europa isolate were very similar. So acid was produced in both colonies as a result of metabolism for 12 of the 14 sugars. Differences here are shown in Halobacterium salinarum producing acid from glucose, but the Europa isolate did not. And the Europa isolate producing acid from glycerol metabolism, but the Halobacterium salinarum did not. And finally, both produced acid through sucrose metabolism, however it was only a weak positive result for the Europa isolate. But all other sugars tested, such as galactose, lactose, and maltose to name a few, all had acid production in their metabolisms by both halophils. So quite a few similarities there. There are also a lot of similarities between Halobacterium salinarum and the Europa isolate when it came to what enzymes were present. However, the results for the hydrolytic enzyme activity varied considerably. As we can see from Table 3, there were 4 different enzymes that were either found in one and not the other, or the other way around. This shows that when it comes to the use of enzymes, there are some differences between these two species. And this could be a result of different material or nutrients available to each in their own environment. For the antibiotic tests, we are testing the sensitivity of both colonies to different antibiotics. The results further supported the similarities between these two species. So 10 different antibiotics were tested, and the only difference in sensitivity recorded was that Europa isolate was sensitive to the antibiotic rifampicin, but Halobacterium salinarum was only intermediately sensitive. Other than that, all the same results between the two. So in the physiological test, we wanted to see how different environments affected the growth of both colonies, and then compare them to one another. So here we manipulated sodium chloride concentration, temperature, and pH. I'll just go through them one by one. Starting with figure 6, sodium chloride concentration, we can see that both colonies maximize their growth at higher concentrations. So 4 moles per liter for the Halobacterium salinarum and 5 moles per liter for the Europa isolate. Both these colonies grow extremely well in high salt environments. Right? We already knew this about Halobacterium salinarum. This was also expected for the Europa isolate due to the high concentrations of sodium and magnesium salts contained in the environment that it was extracted from. Alright, so the results of the effect of temperature growth are also very interesting, but are also as expected. So here in figure 7, we see that the Europa isolate has more growth than Halobacterium salinarum at colder temperatures, but less growth at higher temperatures. Considering the environment they are both found in, this is expected, Europa being much colder on average than temperatures found on Earth, where Halobacterium salinarum are found. And in figure 8, there's not a whole lot of variance in the effect that pH has on growth between the two colonies. So both species have a maximum growth in a neutral environment at pH 7, and their growth declines as the pH gets farther from the value in both directions. This contributes to the similarity between the two species. So, if you remember, the purpose of this experiment was to determine if the isolate found in Europa was halophilic. And we were going to do this by comparing its morphological, biochemical, and physiological characteristics with the halophil here on Earth. After comparing the two, we found a lot of similarities in both physical appearance and biology of the two species. Most importantly though, the physiological test showed two species had many of the same responses to growth in various environments. So first off, the Europa isolate's growth at low temperatures indicates that it's able to survive in cold temperatures that would be found on Jupiter's icy moon, Europa. But most importantly, the Europa isolate showed maximum growth at a high salinity. This leads us to the conclusion that the Europa isolate is indeed a salt-loving halophil. Thank you for your time.